Hey
Uh,
there.
for some
How are
reason,
you?
I got kicked out of Zoom, so I had to rejoin.
Got it.
Um,
Yep.
I have so I redid the section bank exactly
Hmm.
how we talked about redoing it, and I still had a couple questions. There was one passage that I think I did completely wrong because I missed so many on it. So I was hoping we could go through that together, and then I just had a couple of individual questions that I was hoping you'd be able to help me with.
Yep. You can do that.
So I needed, oh, I can't do that. I guess I have to jump through. I just have it reset on it all being blank. And then I can, hopefully I can remember what I went with. So I did really poorly on this passage,
Yeah.
and I was kind of hoping we could do it together because I have like the exact like diagram that I drew for it, and then like the relationships I came up with, but I wanted to see how I could compare it to what you do to see where I went wrong.
Got it.
Sorry, getting notifications. Okay. Yeah.
All right. You could start reading.
Okay. Um, Second messenger camp activates protein kinase A, which can then activate nuclear transcription factor camp response element binding protein CREB. CREB exists as two isoforms in cells, one being CREB 327, 327 residues.
All right.
CREB 3,
Sure.
yes.
So what can you tell me about, so buzzwords, right? So what can you tell me about kinase?
Um, Kinase means it will add ATP to phosphorylate something.
It can, uh, so what class of enzyme is it?
Um, like um a transferase, right?
Yep. So it just transfers phosphate groups. So it doesn't have to always add them.
Hmm.
For instance, like pyruvate kinase, that changes PEP,
Hmm.
pyruvate, into pyruvate. So it's removing a phosphate in that case, right?
Hmm.
And gives it to ADP to make ATP. So
Hmm.
kinases are very high yield because you want to know that they use S, T, Y. So what are these amino acids?
It uses serine, threonine, and tyrosine.
What do they all have in common?
They all have an OH group.
Yep. And when you see an OH group, I want you to think nucleophile.
Uh-huh.
And typically, carbonyl groups are good electrophiles, but a phosphate looks like this, which is somewhat like a carbonyl group, right?
Uh-huh.
So the nucleophile will attack if it's from ATP, the outermost phosphate is the gamma phosphate because you go from the pentose sugar. The
Hmm.
first one is alpha, then beta, then gamma. So
Hmm.
it'll take the gamma phosphate. And so, yeah, that's what kinases do. Now, when they tell you something like this, 327 residues,
Hmm.
expect them to ask you something about the weight, meaning, so you have to know that the average weight of an amino acid is 110 Daltons.
Uh-huh.
And so if they tell you 327 residues and they ask you like the weight of that, you can
Uh-huh.
calculate that,
Uh-huh.
right? So those are some buzzwords. Now we're talking about activates now. So we can, if it said like regulates or something like that, then we don't know in which direction it's going. But activates means that it's going to cause more of something to happen. So we can say that camp would be proportional to pKa, which can be proportional to Krebs.
Hmm.
And what are isoforms?
Like different protein forms.
So, like, tell me more about that. Like, how do they form like differently?
Um, through um different like exons during transcription, like forming different proteins.
Yep, alternative splicing, right? So if you have like exon 1, 2, 3, and 4, you can have something that has, let's say, exon 1, 3, and 4, and then another one that has exon 1, 2, and 4.
Mhm.
All those different things can get you isoforms. Okay, good. All right, continue.
Crab 327 is phosphorylated by glycogen synthase kinase 3, GSK3, at serine 115.
All right, so now maybe I'll write something like um an arrow to like Krebs with a phosphate with a circle around it, a P with a circle around it to represent the phosphate, and that this is done by GSK3.
Mhm.
Okay.
And then pKa phosphorylates Crab 327 at serine 19.
Okay. Oh, PK pKa phosphorylates. Okay, I'm gonna put 119 here, and then I'll put like another arrow with pKa, and then I'll put the phosphate, and then just grab one. Oh wait,
I think.
this was 115,
Yeah.
and then this is 118. Okay.
Um, Variants of wild type Crab 327, Crab 327 wild type that cannot be phosphorylated at serine 115 and 1 or 119 were created. Crab 327 115 and Crab 327 119 respectively.
Okay, so they did something likely to change the sarin into something that can't be phosphorylatable.
Mhm.
Um, and we want to be able to differentiate the pKa that does it at the 119th position and the GSK3 that does it at the 115th position. So like the Krebs 327 115 means that it can't be phosphorylated at that point.
Mhm.
Okay.
Experiment one. Wild type and Crab 327 119 were incubated with various combinations of pKa and GSK3 in the presence of radio-labeled ATP. SDS page gel electrophoresis was performed under reducing conditions, and radio-labeled protein bands were detected. Figure one. And
Okay.
then
So,
expression is phosphorylation of Crab 327 wild type and Crab 327 119.
yep. So the 119 means that pKa um won't be able to phosphorylate it, right?
Mhm.
And so here we see that for the wild type without the mutation and pKa gives you a phosphorylated Krebs.
Uh-huh.
And then pKa plus GSK3 for the wild type gives you, I guess, a thicker band,
Mhm.
which would mean likely that it's phosphorylating, you know, both 115 and 119.
Mhm.
Okay.
And then experiment two, Crab 327 wild type was incubated with radio labeled ATP in either pKa, GSK3, or sequentially with pKa, then GSK3. Isoelectric focusing and autoradiography were used to detect phosphorylation. Figure 2, isoelectric focusing of samples.
What's isoelectric focusing?
Um, I believe isoelectric focusing is when you separate the proteins based off of charge.
Yeah, so you have a pH like gradient, and the protein will move along that pH gradient to get to a pH at which it'll exist as a with no net charge.
Uh-huh.
Hmm.
Sorry, I'm just writing that.
Hmm. So we see control, GSK3, and then pKa. So, and they're doing this for the wild type. So seems like GSK alone doesn't cause this phosphorylation, but pKa does, and that it's further closer to the negative sign, so it has more of a plus charge, I guess. And I
Hmm?
guess since the phosphate has a negative charge, maybe it would be moving down then. That makes more sense. Okay. Oh wait, no. If the stuff is down here, it should be moving up. But anyway, I don't know about that. But let's see. So, and then pKa, then GSK3 gives you something here, thicker band. So it could mean both points get phosphorylated, but and that could also maybe explain why it's not so close to the negative part. Um, okay. So that's what we see there.
Mm, okay. A specific assay uses the percent conversion of chlorim chloramphenicol CHL to its acetylated form what is it? CHLA as an indicator of the transcriptional activity of CREB. Plasmid encoding CREB 327 wild type and its variants were transfected into the cell, and their transcriptional activities were measured. Endogenous CREB 327 activity is not detected. Table 1.
So I just wrote CHLA proportional to Krebs transcription.
Uh-huh.
So that's something that they use as an indicator of transcription of Krebs.
Uh-huh.
And okay, and then let's see. So for the wild type, um, percent conversion of okay, that's the acetylated form. So for the wild type, it's 27%. The one that can't be phosphorylated at the 115th position, 8.5%. Um, one that can't be phosphorylated at the 119th position, 1.7. Control, 1.6. Okay, so it seems like, well, if you can't phosphorylate the 119th position, which means that you can't use pKa and but could use GSK3, seems like it produces a larger decrease. So compared to, if you can't phosphorylate 115. So it seems like the 119th position would be more would cause more of the Krebs A or the CHLA, which means more of the Krebs transcription. So maybe I would just say like, okay, like pKa greater than maybe GSK3 in terms of like Krebs transcription. So okay.
Okay. Um, and then
Mhm.
so the method used to visualize Krebs 327 wild type in experiment 2 must include which step?
Okay.
Um, and
So,
then
yep.
yeah, so we talked about the pH gradient.
Oh yeah. Yep. So they're talking about isoelectric focusing in figure two, and yeah, they're doing that for a protein, right? So you're not going to be able to use PCR on it.
Mhm.
CD,
Um,
yeah, what's up?
I think you could just quickly break down PCR for me because I tried looking it up but on Khan Academy, but I feel like it wasn't like I was still a little confused. Like I understand like you kind of have to amplify.
Yeah, so if you have a small sample of a gene, or DNA, it's like I saw this in like some like forensic TV show, but like back in, let's say, like the 80s or 70s, if someone committed like a crime and you had DNA, um, if they were to sequence that DNA, like they would use it all up until PCR was invented, which lets you take a very small amount of DNA, and amplify it and to find target, sequences. You could also do that uh to find, like, if you have like a virus, if you have like COVID or something, there's like a PCR test that you can do that can amplify, like, the gene for COVID to like see if, any of it's inside you. So it's just a way to amplify some DNA, some
Okay.
target DNA.
The
So
PCR is always done with only like DNA. So if it were to be like, oh, like we have mRNA, what sequencing can we use? We can ever use PCR.
oh, no, no. So that's actually what you're going to be using. Uh, because you're using, like, for like RT-PCR, like you're going to be using reverse transcriptase to
Mhm.
turn. So like if you could turn the DNA into mRNA, then you can use reverse transcriptase to turn that mRNA back into cDNA.
Uh-huh.
So yeah, it'll just involve nucleotides, so that can include RNA, can include DNA.
Uh-huh.
But since we're talking about proteins here, we're not going to be able to use PCR, right? So that's out. And then C says denature the sample so the proteins unfold into a linear chain. Um, well, they would have told us that they would have said like we added SDS or, some BME or something like that. That would
Mhm.
be so for more so for, like, denaturing gel electrophoresis or reducing gel electrophoresis. So C is out. And then
Mhm.
D, treating it with a chemical that'll add an overall negative charge to each protein. That's also something that you do in like a gel. And you know, that could help here, but we are checking to see, like, we're doing isoelectric focusing, so we're not gonna want to give everything. And like if it was a regular gel, yeah, you would want to give everything a uniform charge so that it can move in one direction. But for isoelectric focusing, it'll move in whichever direction it needs to have um a net charge of zero. So, yep, stable pH gradient is what you use for isoelectric focusing.
Okay.
Mhm.
Sounds good. Um, okay. What conclusion about phosphorylation Krebs 327 is supported by the data in figure 1 and figure 2?
Okay, so figure one, we see that pKa can do it, and so okay, GSK on its own can't do it. Um, but pKa can do it, and then GSK plus pKa can do it. And then here we see again pKa can do it. And then here it says pKa then GSK, right?
Mm.
So so we're gonna look for something like that. Um, so A says that GSK3 also phosphorylates at 119. Um, that's not true, right? Because it should phosphorylate at
We
one.
did see a band, right?
What's up?
You'd see like a band here if that were true, right?
Um, 119. Yeah, yeah. Good. Yeah, so okay, that's out. GSK3 phosphorylates at a faster rate. There's no way for us to figure out the rate of anything here. Uh,
Uh,
right? We're just like doing uh like SDS gel electrophoresis, and then we're doing isoelectric focusing. So that just is going to give us like the end number of however many, things got
mhm,
phosphorylated. So there's no way for us to figure out anything about rate. Um,
mhm.
and then it says pKa occurs at more than one residue. That's kind of going to be wrong for the same reason A is wrong, because it's saying, you know, that it, in addition to doing this, it can do it, at a different residue. So
Yeah.
that's out. And then D says pKa phosphorylation happens first, and we see that mostly here. So I think that would be D. Yep.
So
Yep.
when designing var yeah,
Yeah.
this
So
one,
when, yeah. Well, so like, yeah, a lot of times when they do this, for instance, yeah. So the
uh,
glutamate and
yeah,
aspartic acid would be called phosphomimetic. Like,
uh-huh,
like if you're mimicking something, you're like, you know, acting like it. So
uh-huh.
threonine and tyrosine can get phosphorylated, and then they would have a negative charge. And glutamate and aspartic acid, already have a negative charge, so they can behave as if they're phosphorylated. Yep.
Mm. Um, which statement explains how two forms of Krebs can be generated in the
Yep.
cell?
So we talked
Oh
about
yeah.
that too. So yeah, you see how like the buzzwords, right? When you read a passage, that
Hmm.
helped us figure out like or predict what they were going to ask,
Hmm.
right? So this is where I asked about isoforms.
Yeah.
So yep,
Um,
with splicing, yeah.
which statement about Krebs phosphorylation is supported by table one? So this one was actually one that I like really, really struggled with when doing the section bank.
Okay.
Um, and let me see. I I don't know if I still get it at this point. Like,
Okay, so let's go through this. So A says phosphorylation by GSK3 has no effect. Well, it GSK is the 115 one. So
yeah.
if here, this means that it can't GSK can't phosphorylate it, and we see that the percent conversion goes down.
Hmm. But
Yep.
yeah.
So it does have an effect, but
Um,
PKA likely has a higher effect. But yeah, so this is out. And then PKA has a higher specificity for wild type than Preb 115. There's
I think I put this one.
uh-huh.
I went with this one because I thought 115 there was no specificity for that at all. So because isn't that with GSK? So then I was like, oh yeah, like that means that would have to be true because crab it like can do it with the wild type, but then 115 is involves GSK.
Preb 115 here means that it can't be phosphorylated at the 115th position,
Mhm.
which means GSK can't phosphorylate it, but there's still the 119th position that PKA can phosphorylate.
Mhm.
So The thing is, so like the wild type means that it can get phosphorylated at 115 and 119,
Mhm.
it's, GSK and PKA respectively. But this 115 here means that GSK can't phosphorylate it, but it still has a phosphorylatable 119.
Mhm.
So there's nothing that would tell us that PKA has a higher specificity for the wild type. So, but I think what you saw here was like if it's able to phosphorylate the 115th position, but it means the opposite,
Oh, okay. That
right?
makes-
Because yeah.
And then phosphorylation by PK can partially activate Krebs 327.
Yep. So remember when I looked at this and I said that we have PKA a has more of an effect, did it make sense like why that was?
No, I'm still like lost on why that answer choice works. In my head, it was like, oh, phosphorylation by pKa can partially activate, meaning it still has an effect on its own without the GSK. But I don't know if that thinking is right.
So this wild type is going to be able to be phosphorylated at 115 by GSK3 and at 119 by PKA.
Mhm.
And then this 115 one means that it cannot be phosphorylated at the 115, which means that PKA is responsible for phosphorylating this
Mhm.
variant.
Yeah.
And the 119 means that it can't be phosphorylated at 119, which means that GSK is the only thing that's phosphorylating that.
Mhm.
And so that means that if you just have PKA phosphorylating it and no GSK3, we drop from 27 to 8.5, right? But when we have 119, which means that PKA can't phosphorylate it, but GSK3 can, you have from 27 a much bigger decrease.
Yeah.
So that must mean that PKA plays a larger role in, you know, this converting this to you know, the the Krebs transcriptional activity.
Uh-huh.
So Um, so yeah, this one C would be good. D just talks about K-cat, but there's no way we can figure out anything about K-cat here. That's
Mhm.
just the catalytic turnover rate, which determines like the Vmax, but
Mhm.
there's no enzyme kinetics that we're doing here. So I think it'll be C. But like, yeah, does it make sense how I did this? Like if you like this one, 115 means that it cannot get phosphorylated at 115. And since
Yeah.
GSK phosphorylates it at 119, I'm sorry, at 115, it means that PKA is doing the phosphorylation here.
Mhm.
And then the 119 means that PKA can't phosphorylate it, but GSK3 can. And that causes, so that means if you let PKA work without GSK3, yeah, you'll lose percent conversion from 27 to 8.5.
Mhm.
But if you can't phosphorylate it with PKA, but you can with GSK3, it drops all the way down to 1.7.
Yeah.
So
So,
A, yeah, has a larger effect here. And
oh,
the only choice that's closest to that is C.
okay, that makes sense now.
Mhm.
And then under certain conditions, PKA and GSK3 have been shown to autophosphorylate. The control group used in experiment 2, lane 1, was designed to account for this possibility. Given this, the control group most likely contained, and then it says Krebs 327 wild type with ATP, but no PKA and GSK3. Um, so this question really confused me too.
Got it. So, okay. Experiment two, they took Crab 327 wild type and incubated it with ATP and then pKa GSK. So that means that pKa and GSK3, they can take phosphates from the ATP and phosphorylate Crab 327 wild type, right?
Mhm.
Now, if you want to see pKa and GSK3 like phosphorylating themselves, and we want to have it so that it's in the control group, lane one,
Mhm.
we should get rid of the thing that's okay, here and here, the thing that is phosphorylated is the Krebs, right?
Yeah.
So how can we ensure that something like this would be phosphorylating something that isn't Krebs?
Sorry. Could you repeat that?
Yeah. So this one and this one we know will be Krebs being phosphorylated, right?
Hmm.
So we want to see if the thing that's phosphorylated is not Krebs and
Hmm.
either, pKa or GSK3.
Hmm.
So that means that if we got like a band, like let's say here, how do we know that this band is a phosphorylated GSK3 and not Krebs? We don't know, but if we don't include Krebs and we see a band here, that means that GSK3 has to be the thing that was phosphorylated, right? So yeah, so for this, so like okay, if you had this with no ATP, pKa, or GSK3, we're not gonna see any bands because there's no phosphate source, and
Yeah.
and we don't have the enzymes either. So
Hmm.
that's gonna be out. Then C, pKa, and GSK3 with no ATP or with no Krebs. So
It's
this kind of
nothing.
helps us in that we don't have the Krebs,
Yeah.
but since we don't have ATP, there's no phosphate source, so we're not going to see any bands as well.
Uh-huh.
That's out. And then pKa and GSK3 with ATP but no Krebs, that's perfect because that means that whatever band we see cannot be Krebs that gets phosphorylated and must be either this or this, right? And then so A says Krebs with ATP but no pKa or GSK3. So that's just going to show us, I mean, that's not going to really show us any bands because the ATP doesn't have anything to take its phosphate and give it to the Krebs. But also, if we're trying to see if pKa and GSK3 are phosphorylated, we definitely need pKa and GSK3, right?
Mhm.
So, yep.
So I guess the reason I missed this was because when I saw autophosphorylate, just assumed like I just assumed, oh, don't put ATP or Krebs and see. I guess like I guess I didn't really understand how autophosphorylating worked. I just thought like it was like consistently turning itself on.
So auto part is referring to like itself.
Yeah.
So if pKa autophosphorylates, it can phosphorylate itself.
So it has to still take that ATP from somewhere else.
The phosphate from something else, yeah,
Yeah, the phosphate, sorry.
yeah. And like, we know from the kinases that it has STY that takes the phosphate from ATP.
Yeah.
So we know that kinases will autophosphorylate just as part of their mechanism, right?
Mhm.
Before they eventually give it to a substrate.
Mhm.
So yeah, so key here is we want to see if pKa and GSK3 can be phosphorylated themselves.
Yeah.
So that means we need pKa and GSK. We need a source of phosphate, so
Mhm.
ATP, And we need something like that we can't have something that can get phosphorylated and give us a band um, that isn't pKa or GSK3. Like meaning if we had Krebs and we saw a band, we don't know if that band is phosphorylated Krebs or pKa or GSK3 getting phosphorylated. So that's why you don't want the Krebs.
Okay.
Yep.
Um, the next question, I guess I'll just move backwards because 31 was another question I had. The diagram shows the size and positions of exons numbered and introns lines of a gene that codes for hypothetical protein X, which can exist as two isoforms, either 16 or 17 amino acid residues. Which technique can be used to determine if a sample of cells express both isoforms?
Hmm. Okay, so we know that introns will be leaving, and we know that exons will be spliced together in different ways.
Yep.
Um, now 16 or 17 amino acid residues long. So three base pairs makes a codon, which codes for an amino acid. So I'm going to divide these by three. So this is going to be 8 amino acids. This is going to be 3 amino acids. This is going to be 2 amino acids. And this is going to be 6 amino acids. And now 16 or 17. So we can have, if we have exon 1 with 8 plus exon 3 with 2 plus exon 4 with 6, this gives us 16, right?
Mm-hmm.
And then for the 17, we would add exon 1 again with 8 plus exon 2 with 3, and then exon 4 with 6. So that'll give us 17, right?
Mm-hmm.
So, okay. That's okay. So that's what we can use. Now,
I guess I didn't know why we had to like I understand why we broke it down to the codons, but
uh-huh.
I guess the reason I was confused was I didn't realize this was gonna have to do like when I did this problem, I didn't realize that there was gonna be math involved and um so
Yeah.
I guess I just don't understand the basis for what's occurring.
Yeah. So there's always going to be math, always, and
Yeah,
especially when you have numbers, right?
yeah,
So if they're telling us that we have isoforms, and they're telling us that the two isoforms, there's one that's 16 amino acids long and one that's 17,
huh.
want to be able to tell, or so we're given like mRNA here, and we want to figure out how to go from mRNA to amino acids, right?
Mhm.
So that means that's where like the codon comes in, right?
Yeah.
And three a codon is three nucleotides.
Mhm.
So that's what I did first. So I divided, the base pairs of these exons by three to give us the number of amino acids that you would get.
Yeah.
Then I figured out the splicing that can happen that can give you each isoform.
Yeah.
So so that's where you want to start with, right?
Mhm.
And now we're being asked, how can we determine we if a sample of cells express both of the isoforms?
Mhm.
So we need exon 1 and 4 definitely.
Yeah.
And so C has exons 1 and 3,
Hmm.
and that won't give us, they won't give us any of these, right? We need at least 1 and 4.
Yeah.
Okay. And then, well, B is talking about a DNA probe for mRNA. I'm not sure about that. And also, it's just talking about exon 3.
Uh-huh.
That'll show us the one of the isoforms, but not the other. So I'm not sure about that. Um, D says to cut in the middle of exon 2. You don't want to cut an exon. We need the exons intact, right?
Yeah.
We want to get rid of the introns. So that's going to be bad. And then A says synthesize cDNA from protein X mRNA using primers overlapping exons 1 and 4. So that includes the exons 1 and 4, which is what we need for both isoforms. And yeah, that should work, I think.
So anytime that I see something like, oh, here are the two isofers or isoforms we give you, and they give me numbers like base pairs and stuff, then I'll know like, oh, okay, like I wanna like they gave me how many amino acids there are because I have the amino acids, I gotta divide the base pairs by three and then figure out which exons will total up to the amount, I'm guessing.
Yeah, like you want to be very flexible with math because they can ask they could ask a trillion questions here. I can ask you like, okay, um like what's the weight of the isoforms, right?
And then I would divide that. I would divide each by three and then I would multiply. I would get it into Daltons, right? Like
Uh-huh.
100.
Yep. So yeah, so like 16 amino acids, one amino acid, 110 Daltons. You can
Mm-hmm.
maybe even do zero point 0.1 kilodalton or 1 over 10. That's,
Mm-hmm.
could be faster that way. So yeah, they can ask you like all sorts of things mathematically. So you just want to be very, very flexible with anything. There's no like real hard and fast rule, but just be able to be very, very flexible.
Okay. Yeah, I think this one just threw me off because I didn't know that I had to do math when I did it.
Yeah, always, there's always going to be, there always will be math in some shape or form, no matter what.
Yeah. The next one I need help with is, sorry, I'm just keeping an eye on the clock. I needed help with 16, 17, and 19. And then those are the last three that I was confused on the section bank. Okay. Yeah, this one confused me.
Mhm.
Um, because it talks about the ligand-gated ion channel.
Yep.
And so when I saw NA, I was like, oh, action potential. These, like, this one's got to go. And then I saw this and I was like, oh, action potential. But I think the answer choice was C, and I still don't get why it was C because I just assumed, oh, calcium, like calcium is probably going to be the ligand one. So I was just hoping you could kind of explain to me, like, I guess why it was C because it talks about the motor endplate resulting in depolarization. But then when I think of, oh, there's an influx of Na and then there's depolarization, I'm assuming that's electrical.
So you're right when it comes to between like synapses between neurons,
Yeah,
but there's also the neuromuscular junction,
yeah.
which is between a neuron and a muscle,
Uh-huh.
right?
Yeah.
So this is when, so it's related to this as well. So
Yeah.
so in the muscle, our endoplasmic reticulum in the muscle is called sarcoplasmic reticulum.
Uh-huh.
And it we have these T tubules, and
Yeah.
it basically sequesters or holds like a lot of calcium ion. And the calcium ion can get released,
Yeah.
and it can start this muscle contraction process. But what it can do is it can get released, taken up into the neuron, and then the neuron will release a neurotransmitter like acetylcholine,
Uh-huh.
which leaves, and it goes into this neuromuscular, like the uh, synaptic cleft between the neuron and the motor end plate of the muscle.
Yeah.
And so that the ligand is going to be the acetylcholine. And in order for us, we're still having an action potential,
Yeah.
but it's going instead of from neuron to neuron, it's going from neuron to muscle.
Yeah.
And in order to have that happen, you need that neurotransmitter to act as the ligand. So when it binds to the ligand-gated ion channel on the motor end plate, it can let the sodium enter.
Uh-huh.
So, and acetylcholine, whatever excess we have here, is broken down by acetylcholinesterase,
Mm.
which can then have it get reuptaken into the neuron. So I remember like when I was like maybe 8, there was a movie I really liked called The Rock, and it was about VX gas, which is like sarin. Have you ever heard of like sarin gas? Or so basically so it's like a nerve gas, and
Yeah.
basically what it works is how it works is um it's an acetylcholinesterase inhibitor. So
Hmm.
acetylcholinesterase takes the acetylcholine and breaks it down and puts it back into like the neuron. Uh,
Yeah.
but we know that acetylcholine is what's required for the muscle contraction.
Hmm.
So if you inhibit the acetylcholinesterase, you don't have anything breaking down the acetylcholine, which means you just have excess acetylcholine causing contractions, and the contractions happen so fast, like you're twitching, and and especially for like your diaphragm and stuff, like you lose control of all of that and you convulse and you die.
Oh.
So yeah, you can always learn stuff. Well, I learn stuff from movies all the time. But anyway, yeah. So you have that right idea,
Uh-huh.
this, right? Um, and that would be the voltage gated channel. But
Yeah.
yeah, for the neuromuscular junction, it's different, and that's
So
calcium.
because it's talking specifically about the motor end plate, then I'm going to be like, oh, okay, motor end plate, this is tied to muscles. There needs to be some type of neurotransmitter
Mhm,
that's attaching so there could that NA can enter.
mhm, mhm.
That makes sense. Uh, so this I was confused about because I've gone over blotting and or Southern blot in the past when I took that MCAT, but I don't remember it. It talked about like the technique of it, and I did this on. I think I was on this question right after I met with you on Monday.
Mhm.
So it's a little hazy. I'm a little hazy on the terminology, but it was talking about how there's something like in the middle where it needs to cut like a specific sequence it notices or something, like
Yeah,
some type of sequence. And so I guess I was just a little confused because I went with one on the end and it was like, oh no, it can't be that one because it needs to be from the middle. So I just kind of wanted a little bit of clarification on how this works and why I
yeah.
don't know the answer choice either, if I'm being honest.
Uh-huh. Yeah. So in general, like, what we can do is we can like look at where there's, a difference. So here seems to be one, and then here's another one, and then
Yeah.
there's another one, and there's another one, right?
Yeah. So I think I went with like when I was taking this, I was kind of I was moving fast, so I just clicked B because I was like, oh, I see a change there right on the end. So I went with that, and I just wasn't sure why it needs to be in the middle, I guess.
So two things. So like, because like I don't know if I said this, but I always want to give students more than one way to get your answer. And, and then a lot of times it involves what if I don't know? Because it's not so about knowledge as much as, critical thinking. So right away, you should see that one is different from the rest,
Yeah.
right? And which one is different?
Um, this one.
Hmm. Right? Because like, okay, we could say like, okay, these two are similar in that they're at the end.
Mhm.
This one is at a different end, so it's not as similar as these two. But then again, this one is the most dissimilar. It's the most different.
Mhm.
So without any questions, like, if you don't know anything about this, then you should choose the one that's the most different, right?
Mhm.
But now let's actually talk about it. So when you're doing any type of this blot stuff, right? You need to, first of all right, so you need to use a restriction endonuclease.
Mhm.
And so endonuclease, so nuclease, what does that like mean, basically?
It's gonna make a cut.
So just the word would mean it would break down a nucleic acid, right?
Yeah.
Like a protease would break down a protein,
Uh-huh.
a lipase would break down a fat,
Yeah.
right? So, so okay, a nuclease would break, or a nucleotide, and
Mhm.
then exonuclease would break at the ends or
Uh,
the outside, because exo means outside, right?
yeah.
And then endo means inside, so that would break something in the middle.
Yeah.
And you want to break something in the middle because you want to break something at a palindromic sequence.
That's the term he talked about. I've never heard of that.
So, what about the word palindrome?
Uh, don't think that sounds familiar to me either.
So, this word forwards is the same backwards.
Oh, yes, I recall this. Yep,
Yep. So
elementary
that's
school.
yeah. So that's a palindrome.
Mhm.
And a palindromic sequence will be complementary to itself reversed, right? Which must be the case because you need to have a 5-3 uh, complementary to a 3-5.
Yes.
That's why it being complementary to itself being backwards is important.
Mhm.
So, for instance, here we have, so like let's say that we cut at this palindromic. Okay, here we go. A G C T 5-3.
Mhm.
What is it? 3-5, right? TTCGAA, right?
Yeah.
So this, so the palindrome of this sequence is complementary to it to itself,
Yeah.
right? So that's a palindromic sequence, right? Now let's say that we cut at anywhere, right? Let's say here, right? Could be really anywhere. So
Mhm.
let's say we cut between a C and a T.
Yeah.
That means we're cutting between the C and the T here as well,
Uh-huh.
right? And that means that we're going to get this segment and this segment,
Yeah.
right? Like this
Yeah.
and like that.
Uh-huh.
Now this means that these overhangs are called sticky ends.
Yeah.
And this means that we can cut, like a plasmid or something, um, like a circular DNA, like a plasmid. and we
Uh-huh.
can insert a gene in there. So like insulin, before we did anything like this, you would have to get from like a human. Now that's obviously hard and whatever. So what they did is they take the human gene for insulin, they put the gene inside of they took like a bacteria with a plasmid, circular one. They cut the plasmid open and oops, like this, and then they take our gene for insulin, and then
Yeah.
they just throw it in there, and this gets to close up again.
Yeah.
And that's how you can put, a gene into something. Uh,
Mhm.
so yeah, so that's essentially what they're doing here. You know, um, so single point mutation causes a non-functional protein, and which one most likely? Okay, yeah. So this is like, so it's just the fact that they said Southern blot. That means that we're going to be cutting open the DNA
Mhm.
with like, yeah. So yeah, so essentially we don't know like anything about it. It's likely going to be the answer that's the most different. And then the reason that it's A is because, and it's at the middle, is because you want to cut at a palindromic sequence. Because if you cut here, right, you're just going to get like these blunt, I think they're called blunt ends,
Yeah.
and they can't stick together if they're blunt ends. They need to have the overhanging sticky end so it can stick back together. But yep, that's the idea. So
Oh, okay.
yeah, so a palindromic sequence, a palindrome is a word that is the same forward as it is backwards.
Yeah.
You want a sequence that its forward one will be complementary to its backward one. And you want that because if you make a nick, you cut, you're going to get your sticky ends. And that means that you can do whatever you want. Yep, there we go. Sticky end overhang. And then blunt end, no overhang. So there's no way for this to connect back to it.
So you do the middle because you want those sticky overhangs so that way you have like a way to like connect the DNA back together.
Yep, because if we had these blunt ends, how would we connect these, right?
Yeah, we wouldn't be able to.
Right? Like
It has to be the
technically there's double strand breaks and things that can fix that. But
yeah,
that would have to be fixed by some enzymes. But
yeah,
sticky ends are just the G is naturally going to bind to the C through the hydrogen bonding, right? So
yeah,
this doesn't even require an enzyme. It can just naturally stick back together. Yep.
yeah. Okay. Sounds good.
Mhm.
And then the last one I was confused on was 19. But I don't know if we need to read this. I don't know if we need to necessarily read this because when I was going through it, it said that it could be looked at as a standalone, which made me more confused.
Mhm.
Um, it said a vDNA sequence encoding a protein is inserted into a host genome by in. The protein is translated from the hypothetical mRNA sequence based off of the passage the segment of the original viral DNA had what nucleotide encoded.
Okay, well,
And then I remember the answer choice, the correct one is A. And I remember that because it had to be the exact same thing, but
yeah.
I didn't know that.
Yeah, so I was gonna say like almost nothing is really totally standalone because here they're saying that this is a HIV. Okay.
Yeah.
HIV is a retrovirus, which means that gonna turn it's gonna turn mRNA into DNA. So because if I read this as a standalone, I would see vDNA and I would get rid of these,
Yeah,
right? So yeah, so be careful about anything being like standalone because
yeah,
the passage is the only thing that really tells me that this is HIV, a retrovirus. So that means that it's gonna be like not thieves, not
yeah.
the ones with the thymines, right?
Uh-huh.
And then um okay. So basically what happens is the 3 to 5 part gets a primer added, and then it fills out this end to
Mhm.
make that template. So this would be the coding strand, and this is the template strand, and then this will be, well, it's backwards, but it still would work. So basically, this one is the original coding strand, and 3' to 5' would be made, and that would be the template strand. And then this mRNA would be made from the template strand, being complementary to the template strand.
So it's asking for, like, basically it's asking me to like talk about what like the original strand that made like 5 prime GG CAA.
Yep. So like let's say we had DNA like 5' to 3', 3', 5',
Yeah,
and we want to make RNA. We're going to make we're going to take, so this 5 to 3 is going to be the coding strand,
yeah,
and complementary to that would be the template.
yeah.
And then complementary to the template would be the new mRNA strand.
Uh-huh.
And since the coding strand is complementary to the template and the mRNA is complementary to the template, that must mean that the coding strand and the mRNA strand are the same,
Mhm.
right? And then yes, this can be turned into cDNA using reverse transcriptase. So we're essentially going backwards like that. So we have the mRNA, this part here,
Mhm.
and we're essentially coding for the cDNA part of that, which is just going to be the same as that.
Okay.
Yep.
So basically this is saying we've got our 5 prime to 3 prime N, and then we've got our 3 prime to 5 prime N. So one is the coding strand, and then one is the template strand.
Mhm.
And then so it gives us one, and then it asks us to basically come up with the other.
So yeah, so your coding strand will be the same as your mRNA strand,
Yeah.
and and that's just because the coding strand complementary to that will be the template strand, and the template strand is the one that the mRNA gets made from, like, complementary to. And so since they're both complementary to the template strand, they're going to be the same. Yeah.
Okay.
Yeah.
That makes sense.
And yeah, and it's only has uracils in it just because it's HIV.
Okay, yeah. So we that's like so we needed to know for this question that HIV one was.
Hmm.
Okay.
Reverse a retrovirus.
Yeah. Okay.
Hmm.
Perfect. Well, that was everything that I just wanted to review before I take my practice exam tomorrow.
Awesome. Okay. So let's see. Which one are you taking?
Tomorrow I'm taking full length two from because remember I got a little behind and then I also took
Full
uh,
length to like blueprint, was it? Okay. And you're using what for cars?
Jack Weston.
Okay. Got it. All right then. So yeah. Any other questions or anything? Okay. So yeah, it should just be 87 for today.
It's selling 87.
Mhm.
I'm gonna send you 870.
That would be fine. No, I'm kidding. Um, so yeah. Um,
I have just sent it over.
okay. Yeah.
And
I was gonna say, yeah, keep an eye out for those buzzwords because you saw in like the passage that we did today that they asked us about that stuff, right? So that can help you
yeah,
predict certain things. Yep,
yeah.
got it. So, all right, awesome. Good luck tomorrow. Let me know how it goes, and then I'll see you like probably like next week or something, right?
I think I'm on your schedule for Monday because I'm gonna do my blind review and then just meet with you right after the exam.
Got it. All right. Good luck. Okay.
Thanks, Shane. Bye.